இருக்குங்க சார். Good afternoon, everyone. Uh, Thank you, sir, for the introduction, and it is always pleasure being here at TNIU as an alumni. Uh, and I'm just here to give a brief overview on the work that we are currently doing at NBPGR. This is the overall presentation outline. First, I will give one overview on germplasm variability. So this will help us understand why we do on genetic resources, what is the importance in going on with the genetic resources collection and conservation, why we need to handle thousands of accessions, what is the necessity, because it involves more labor, manpower and long-term conservation, which involves more energy, and we spend much money in it. So whether it is worth spending that. So first we will have an overview picture, broader picture on germplasm variability, and then we will go into the actual omics tools and what are all the possibilities and potential pathways that we can use these omics tools in a much resource efficient way to understand the genetics for accelerating the crop improvement. So here in that I will be using some 4-5 case studies from my own work which is going on at NBPGR, and also I had opportunity through DBT funding to work for 2 years at University of Queensland, Australia. So where I worked on the C4 in wheat grains, and after coming back we took forward the possibility of that C4 in rice, but we couldn't find that. So we alternatively found that there is a implant of photorespiratory bypass which is happening in rice grains. So understanding that genetic mechanism will help us to generate C2 rice in future that will give higher biomass. And same ways with the piperin pathway. So this will give us one advantage on how we use transcriptomic studies even when there is no variability. Like for example, if we take capsicum, we have variability from zero pungent pepper capsicums to high pungent pepper. Whereas in case of black pepper, we don't have such variation. So how we will be using the existing such kind of crop without variability for piperin content to understand its biochemical pathways. And then large scale genomics like genotyping using GBS strategies, how it will be helpful in a resource efficiency. So now coming to germplasm variability, this one slide will view on the evolution of rice and the extent of variability. For example, if you see here, Oryza rufipogon is the wild and also Oryza nivara is the wild. But Oryza nivara is an annual form of Oryza rufipogon. And if you see that circle, the larger the circle, the extent of variability. And during domestication process, this Oryza nivara is evolved to indica type and Oryza rufipogon is evolved to japonica type. That is the concept of domestication in evolutionary process of rice. And here if you see, although the extent of diversity in Oryza nivara is lesser when compared to Oryza rufipogon, from which the annual type has evolved, but if you see the domesticated indica types having a broader variability when compared to japonica types. It is mainly because we have domesticated and evolved different types of rice land races for different purposes. So during the domestication process, the domestication syndrome, what we study and the linkage drag. So this help in getting diversified the traits. So for example, if a wild type has 10 traits in one single plant, it is diversified into each trait for each germplasm land races. So if we want to conserve actual land race, we need to conserve 10 accessions, 10 population, whereas the wild race only one population which is carrying all those 10 traits can be conserved. So this is the extent that wild relatives and wild forms or land races have a larger extent of diversity within the existing population, and as we improve it for cultivars or for commercial cultivation of the land races, their diversity is slowly reduced. There is no kind of a balance, it is obvious. So we need to take that as a domestication cost. So there is no change in that, and with that understanding we conserve the germplasm variability for proper use. Now getting into the transcriptome for the omics tools, first we will see what is transcriptome. The transcriptome is initially coined in 1996 by Charles Offray, and at the time transcriptome means it is less than 5% of the genome, because we have mRNA, tRNA and rRNA, and among these all RNA families, mRNA is the one which is called messenger RNA, which takes the information into the product, but that is having the least representation among the all RNA families, but still that is what we called as transcriptome in initial days. So in this initial days, when we call as transcriptome, it is just a qualitative set, means the complete set of genes that are transcribed in an organism across all developmental stages and at all tissues, all organs. So this is called a transcriptome in earlier days, but later it has had a direct shift from the qualitative perspective to quantitative perspective. It is initially when there is a dot blot technique, when southern blot came initially, then northern blot came. So during this northern blot or dot blot technique, they started quantifying the transcriptome. So then it is from where the qualitative to quantitative shift had occurred with reference to transcriptome, and later there were many further progress in science that lead to a definition transcriptome in its quantitative part. Like for example, if we take arrays, all those dot blot techniques is called as macro array, because in dot blot technique we hybridize the RNA of the sample onto the membrane, and we use probe for washing and hybridization, and later through advancements we entered into microarray, where the probes are hybridized onto the membrane solid surface glass slides, and we used samples to bind, hybridize and then get it cross verified for the presence or absence of genes, and hence they are called as microarrays. So to be clear, if at all it is RNA samples which are spotted, it is macroarray, and if we have probes spotted on the glass slide and we use samples for washing, it is microarray. And parallelly along with this microarray technology came up the real-time PCR. So actual quantification technique along with PCR. So if we see the development, the microarray is the very short-lived, very shortest lifespan among the progress, because it is very narrow between the microarray and the development of real-time PCR. So with real-time PCR quantification rate than when compared to microarray. So that's how the technology was short-lived. But later when GBS came, we used the SNP markers to sustain that. So that is entirely from the availability of GBS. This has taken an entirely new shift, and now what earlier they defined a transcriptome, now is transcriptome, because this transcriptome atlas means it is now the complete set of genes that are transcribed in an organism across development at different tissues or organ levels. So how that, so one example in wheat, and we all know wheat is a hexaploid and it has 3 subgenomes A, B and D. So if specific genes, there is a genome specific expression is there, B and D subgenome are silenced, and here in opposite side it is A genome is totally silenced, only B and D expression is there. Similarly for other 2 subgenomes, and the central triangle with the grey shade is the set of genes which have a moderate expression for all the 3 subgenomes in wheat. So suppose if you want to make a study and if you want to use it for a specificity, so it is always better to use genes from this grey region of the triangle, so that it will have uniform expression across all the 3 subgenomes. So that would be a good reference gene for our quantitative studies. And similarly, this will also give us an idea which kind of subgenome for which kind of genes had a biased expression than a normal expression sets. And another set is, this is an example from rice. Nowadays, the single cell transcriptome has become more advanced from in plant systems as well, other than cell culture studies. So here, due to microatomic sections, where we can take individual cell layers and individual cell type within the tissues. So each and every cell, they make transcriptome analysis and what kind of transcript express that particular cell layers has an implication on the organ development. So this will give an overall picture. So such kind of large scale advancements across interdisciplinary level, like this involves more cytology and to develop a cell specific transcriptome, will really give a shift in defining a atlas. It is a redefined terminology where the which are expressed in a system across all the developmental stages. Whereas transcriptome means it is a quantitative aspects. So that redefinition came because of the possibilities of growth in NGS strategies and other anatomical tools that are available. And now we will take forward with individual case studies of how we used these NGS tools in crop improvement programs. So this one is the C4 pathway in wheat, which was discovered during my work at University of Queensland in Australia for 2 years through DBT funding program. So I just briefly give one example here. So there we used 35 genotypes of wheat from across the globe, collections from India, from Iran, from different regions. They collected and they had a set of 35 genotypes or cultivars of hexaploid wheat, so that we used for studying the grain transcriptome. So how the wheat grain transcriptome has a changes during grain filling. So this will help us in understanding the grain transcriptome, grain development from grain filling as such. So there we had data for early and late grain filling. Early is at 14 days post anthesis and late is 30 days post anthesis. So in two stages we had transcriptome data generated for all these 35 genotypes at the same stage, and these all 35 genotypes were used for study. So these 35 genotypes will indirectly act as a replicates, 35 replicates. So we compare between early and late so that we can understand the complete grain developmental process. Here we took 14 and 30 mainly because these two are the crucial steps in the grain developmental process, where at 14 it enhances the grain filling exponentially, and at 30 it is the about the completion of a grain filling and leads to physiological maturity. So these two phase we took it and then we studied in detail, and during our study we found that all the C4 pathway genes are differentially expressed during early phase. So this gave us a surprise with wheat being a C3 plant. So when we took all these 6 genes and when we mapped, it gave NADME subtype C4 photosynthetic pathway, which is accomplished in wheat grains. So this we were surprised, having known that wheat grains don't have crans anatomy, and we know that there are possibility of C4 only with crans. And when we looked into the literature, then we found there are reports in 1970s and 80s that in barley and wheat they did labeling experiments and showed there is a C4 pathway in grains. But having said that, without Cran's anatomy, they said if and with leaf having C3 and grains having C4, it is possible for barley and wheat to have a C4 in grains. That's what they told. But at the time, it was a dogma that Cran's anatomy is a must to have C4. So after that, there was no further research in those angle from that perspective. But when we did transcriptome, we identified that, and when we did literature, we found everything is there already in 1970s and 80s. So that we pulled all this data and then we studied how this transcriptome can help us in getting that C4 pathway accomplishment. So when we identified all the number of gene copies, we have multiple copies for each of these gene, and this is normal because we have C3 specific copies and C4 specific copies, because C4 pathway itself is a neo-functionalization event wherein the existing gene specifically had evolved through directional selection for accomplishment of C4 pathway. So here we distinguished between C4 type and C3 type based on the targets. Like for example, if we take aspartate 18 genes for this in the wheat hexaploid. So if you see the cytosolic ones of from chromosome 3 is a C4 type, and mitochondrial targeted gene from chromosome 7 is also C4 type. Rest all are C3 type. So based on that organelle targets where the signal peptide shows. So based on that homology, we could distinguish for C3 type and C4 type. Whereas for pep carboxylase and alanine aminotransferase, both are cytosolic. So we could not distinguish there. But however, in case of pep carboxylase, there is a drastic difference in expression between photosynthesis. Based on that, we could distinguish, and it is supported and validated with the single amino acid substitution, which is associated with its functionality for feedback inhibition of malate. So based on that, we identified this from chromosome 3 as a C4 type, and from normal from chromosome 5 as C3 type. Whereas in case of alanine aminotransferase, across these both cytosolic genes, both didn't have any significant expression across the tissues and also across these two genes at different tissues. So we didn't identify, and also we didn't bother much in of that one. And after when we saw that, that is where we started from that point, that it is there, but how it is there and how it is expressed. So after seeing that, we had framed a series of questions. What are all the possibilities, and if we address these questions, we can answer whether C4 is there or not. So when we looked into the literature for each and every questions, for each and every questions there were reports available already, but they are all in 1970s and 1980s. After that reports, so we compiled all those reports and our study, and then we took forward this analysis, and we found that C4 having a specific expression only in grains for C4 copies alone. Like for example, if you take this panel B, we have this higher expression. So even in C4 plants, it is mentioned that there is a significant 100-fold higher expression for the specific C4 copy in C4 plants, but not in C3 plants. So by seeing this, we also wanted to study whether it is same case in leaf or it is a different when compared to wheat grains. So this data is from wheat grains from our own study across all the 35 genotypes. So when we wanted to look into it, there were transcriptome data sets available in public domain for both leaf and grain tissues, approximately 12 dpa, which is only 2 days difference. So we used that public database and we looked whether it is a difference. So when we saw here, you can see from panel A for pep carboxylase, there is very minimal expression for the C4 specific gene in leaves, whereas it is very highly expressed in case of grains, developing grain, different stages. This is at maturity. So if you take here, similarly for that particular C4 specific ones, in grains we had more than 100 folds higher expression when compared to leaves. Since it is also from public database, from data available in public domains, so we can directly say that all these expressions are true, and it is validated because our own data also showed the same results, but since we didn't have leaf specific data sets, we used public domains. And after seeing this impressively, so we collected all the literatures addressing all those questions, and we proposed a model. Since it is all the 6 genes, when we mapped against all the 3 C4 specific photosynthetic subtypes like NADP ME type, NAD ME type, and PPDK, PCK type. Okay, these 3 subtypes are there for C4 photosynthesis, and here when we mapped for all the genes that were expressed in wheat grains, we found that it is a type of NADME subtype. So at the time, based on that, we found anatomically also in 19 published that there are two photosynthetic cell layers in wheat grain. One is cross cells and one is tube cells. These two are the photosynthetic one. So we propose that these two mimic the mesophyll and bundle sheath cells so that it gives a C4 pathway. However, there is a parallel C3 pathway is occurring within each of the photosynthetic cell types. This is what we proposed. And initially, when we proposed within 1 month, commentaries came which were published in Plant Physiology, stating that it is not possible for these evidences cannot be sufficient enough to take forward. So at the time when we approached the same Plant Physiology editor, they called for open argument from our team and their team. And they said after combined arguments it will be published if there is no further questions. After arguments, all the four arguments were combined and published in same Plant Physiology journal within 1 year. So it is from that poor evidence they told it is one of the outstanding puzzle for understanding C4 pathway in wheat grains. Because we also have validated uh, pulse chase labeling experiments, which is a gold standard experiment even considered today for the understanding of the C4 pathway mechanism. So since it is there, we also proposed and then still it is in kind of taken forward. And after that we saw what are all the other researches that say about our which are in line in the progress, research progress. So when we see this is one species, aquatic species, it is Otelia alismoides. This is the first report to have leaf photosynthesis. It is also NADME type. So mesophyll cells and epidermal cells which were photosynthetic, they mimic bundle sheath and mesophyll cells and they accomplish a C4 pathway. So this is very low and it has a low efficient and it is an NADME type C4 pathway. And similarly, uh, we all know sorghum is a C4 plant. It is a direction from C3. When based on the evolutionary concept, always the later evolved developmental stages should also have higher efficient ones in case of directional selection. The panicle which is having use, so which is in conflict of the system. So relationship, like for example, if this is the same orthology of panicle with wheat panicle, that spike, which have less efficient pathway, also any see, based on that this is we for value we propose, uh, normally presently the concept is it has evolved at 24 to 34 minutes but based on non-Fourier photosynthesis which earlier is less effective leaf serial photosynthesis because it is a complex which is all over the across speciation events. So species of itself will act as a representative to understand very. So our analysis we report there is a shift in the evolution of function S4 from this 24 minutes to somewhere between 51 to 55 to 75 minutes. So this will also address the like for example, they see the geological events at 55 million is drastic reduction in CO2 levels, atmospheric CO2 levels. So this will address that particular question because they questioned if there is a drastic reduction in 64 million years, why it took 30 million years for plants to evolve? for C4 pathway. So that gap is unaddressed till date. But when we push this one to the particular non-foliar type, this addresses that particular gap, so that it evolved during the similar time of global reduction in CO2 levels, and upon its post-optimization of evolution, it gave a robust and efficient C4 evolved in leaves. Now we can have a question why it is evolved in grains but not in actual foliar photosynthesis, because foliar it is evolved there. So that one possibility of answer is in case of non-foliar photosynthetic, it is during the grain filling, the temperature in the mid region gets drastically enhanced. So due to which there is a need for an alternate photosynthetic mechanism for having a higher grain filling for healthy seeds. So due to which it would have evolved in non-foliar than in foliar types. Because by the time it gets grain filling, all the leaf have loses its photosynthesis. So only translocation of the photosynthesis happens from leaf. So due to which there would have been the alternative mechanism where they evolved first. And now coming to second case study. Here, forward work I have done in Australia. After coming back to MJ again, I thought it is in wheat. Then it's in Poaceae we have 12 subfamilies, 3 basal subfamilies, and BOP clade, and then Pac-Man clade. So this Pac-Man clade is the one where we have rye, maize, sorghum, sugarcane, all C4 crops are there. And this BOP clade is also called as a cool season grasses, and where the rice, bamboo, wheat come. So rice is Physidae and wheat in wheat family, bamboos are in Bambusae family. So what we thought was this monophyletic origin in the OACA skill. If in OACA it is there, then there actually that non-foliar photosynthesis originated within this BUP clade. So that we want to address. So after coming back, we started working on it. So at the time, I am interested in rice. So for that, we took rice transcriptome analysis and grains, and then we studied. And we looked into the literature. Anatomical part. They mentioned that the cross cells, tube cells, however, only cross cells photosynthetic, not tube cells. So from the evolutionary perspective, from Origa to Tritium, there is a evolution in making tube cells photosynthetic. If we identify exactly in which species the connection had happened from non-photosynthetic tube layers to photosynthetic tube cell layers, will be much helpful in identifying exact origin of non-foliar C4 photosynthesis. So based on that perspective, we want to study in rice how it happens. So I will just give a brief overview here. And when we studied only 2 genes of C4 pathway have higher, not all the genes. So then we put questions. If that is the case, then what other mechanism is there present in rice grains? So when we identified the next alternative study point is photorespiration, because it's a photorespiratory loss. And when we identified all the photorespiratory genes, we had some interesting findings that I will show here based on our. So if you take overall